David Tower, and welcome again to the Theories of Everything program on Channel 31 Community Television. Viewers, this is the third program in our series Theories of Complexity. In our previous program we examined the characteristics and nature of complex systems, what makes them different from simple linear type systems. We also examined the drivers for understanding complex systems better in our environment, in our world and in our reality. In the previous program we pinned down a number of critical characteristics. First of all, complex systems contain a large number of components, as you'd expect. But it's not just the number of components that makes a system critical, it's their interconnection, it's the structure, it's the level of interaction between those components. So we find that the structure, or the networks established that link those components, that link information flows, are, if anything, more important than the components themselves. And those interrelationships established by those networks of information flows are usually non-linear. They're not simple proportional relationships, inputs directly proportional to output. They vary. They vary at the beginning of the relationship, they change midway through. They have to rise to a certain threshold before they have an impact. For example, a plant in its environment, it receives rain, it receives nutrients, but its growth is not straight proportional to the inputs of that water or nutrients. It has to reach a certain threshold before plant development will occur. So non-linear relationships, constantly dynamic, evolving relationships, thresholds, feedback loops, these are the characteristics of complex systems and complex processes. If we start at the level of the cell, it's a good place to start because the cell itself is a complex system. It contains thousands and thousands of different molecules and different processes are running concurrently, hundreds, even thousands of different processes. More complex than any manufacturing plant, in fact. In fact, a number of research institutes have been set up. I mentioned one last week that studies the science of complexity, but many have been established in the last decade to understand and progress bioinformatics. And some have even established virtual cells. These are simulated cells in a computer that try to replicate some of the more basic processes. One of these projects is the eCell project. It has 127 genes in its simulated cell. Unlike the human genome behind me, the map of that contains 3 billion bases, around about 30,000 to 40,000 genes. Each of those genes or combinations of genes can produce hundreds, thousands of different proteins. And so, in fact, around about 100,000 proteins. There are constant feedback loops, genes being turned on or off depending on on which products are being produced. Interactions not only with proteins but lipids, complex sugars, etc. Different functions of proteins. So these little simulated cells are only a beginning. But they do basic things. They take in food in the form of sugars, they process that food to produce certain products that are essential for the internal processes, the internal metabolism of the cell, and produce waste products such as lactose. But again, the whole object of this is to start mapping the networks, the cascading interactions that occur in these cells at the cellular level. And one of the most important starting points for this is in the signalling systems of cells. Now, a cell has to maintain contact with other cells. It has to understand what its role is at any point in time. It does this through messenger molecules that lock on to receptor molecules on the outside of the cell. Without this flow of intercellular information, cells just die, the whole animal or plant dies as well. So this connection is very important because again that triggers a whole series of cascading processes within the cell and creating the various products that cells create and keep us alive. Now, scientists are constantly looking at these interactions, but again, they've started to map them mathematically. They've started to map the interactions mathematically. Remember, in complex systems, the structure is the vital thing. The components are almost secondary, the individual molecules. Obviously not unimportant, but secondary to the relationships between those molecules, to the topology of the networks. For example, one class of messenger cell are cytokines. Cytokines lock on to receptors on the outside of a cell and are vital in the number of immune processes and wound healing processes, for example. Basically, it tells the cell, the messenger tells the cell to replicate, to take care of the problem, when to replicate. Now, that docking process is vital, but by understanding the network flows inside the cell, scientists have begun to realise that it's just as important to know what happens to those cytokine messenger molecules internally. For example, they can be annihilated by other molecules called proteases, but it's important for their robustness, for the cytokines to replicate more, to be recycled. If they're stopped in their tracks, they're not as effective, no matter how well they dock. So understanding that cellular, intracellular process has created the possibility of being able to improve the whole technique of processing cytokines within a cell, and that produces better outcomes for handling diseases and healing wounds and so forth. Now, viewers, the process, the complex cascading of signal processing in life is vital at all levels. Even down at the bacterial level, virus level. There are a number of processes now that are just beginning to be understood because of this broader type of analysis that's being done, this more holistic analysis for complex systems. One of these processes has been studied for a long time, chemotaxis. It's how bacteria find food sources and zero in on them. And it's been understood for some time that there are proteins involved in each of three stages, proteins that involved in picking up the odour of the food source, proteins involved in rotating the little flagella that pushes the bacteria along in a particular direction, and three, the signal processing proteins that keep everything together in terms of information. Now, how those interrelated and how bacteria seem to be able to zero in on their target no matter what the concentrations of the food source are has not been understood, but through a series of elegant mathematical equations using nonlinear differential equations, this process is now well understood, and it happens that basically the bacteria follow a food or odour gradient, and when it finds a particular odour, the molecule will lock onto the bacteria or lock onto the receptor. That will trigger the production of proteins. Particular genes are expressed, proteins are generated, and those proteins affect the way the flagella rotates. Normally, a bacteria moves in a zigzag direction with periodic tumbling in between. When it locates a food source, this tumbling diminishes and it seems to go much more in a straight line. Well, it's the proteins that are generated when the food molecule locks onto the receptor of the cell that does the trick. And they interfere with the tumbling motion, so the bacteria tumbles less and it goes towards its target in a much straighter line. But if it veers off in the wrong direction, the tumbling will start again. The gradient, there'll be a feedback loop, in other words, that shows that the food is becoming, or the food odour is becoming less and less. And again, it will start zigzagging, tumbling to find out where that food source is. Again, through a process of understanding the feedback loops, not just simple linear processes, this puzzle has been cracked. Even things that one would assume have been solved a long time ago, like understanding what sort of molecules switch on in plants at the tip of a stem, the signal to produce a bud or produce a fruit or produce a flower, produce a leaf, how that works, how those signals work, and how the plant development is progressed from those signals. That has not been understood until recently. Again, by understanding the proteins alone and the gene expression, in the case of bacteria, of course, the genes can vary slightly. So amounts of protein produced can vary in concentration and even in type. But these processes appear, because of the structure, because of the networking, the feedback loops, particularly robust in achieving their ends. And this is a very good sign for evolvability. In other words, if you've got a flexible structure that can ride over small variations in biochemical processes, you've got a chance, that organism has a chance to evolve and adapt much more easily than if it's tightly constrained. Viewers, if we move up the scale of life, we come to some very, very complex systems indeed. We come to you and I, but driving you and I is the human brain. And all mammals have very, very complex brains. Ours is one of the most complex, perhaps not the most complex. Dolphins, whales have very complex brains, and it's difficult to decide to measure that complexity. Regardless, though, in our brain, which is about 4 times the size of early hominids, roaming the earth about 4 million years ago, there are 100 billion neuronal components, different types of brain cells, as well as straight neurons. And they can be connected in trillions of different ways, and are. We can look, in fact, you could say the human brain is the most complex object in the universe. It's far more complex, for example, than galaxies, which may contain our own Milky Way, 400 billion stars. We live around one of those stars, but basically a galaxy differs from a living organism in terms of complexity, because the living organism, first of all, has purpose, has teleology. The things that it does are designed to keep it alive, to help it survive, to help it evolve. So it has to have extra structure, extra complexity to do that. It has to have feedback capability to continuously improve its ability to sense its environment, to process information from that environment, to store information from that environment in the form of memories, etc. It has some very complex subcomponents, such as the neocortex, which provide the ability to do abstract thinking. All higher primates have very complex brains, as of course do, as I mentioned, whales, dolphins, etc. The human brain, or any brain for that matter, any living organism's brain, particularly at the higher level, has history. It has, if you like, evolutionary depth, logical depth. It's had to continue to evolve over time. So it's developed the ability to compartmentalise different functions. It's developed redundancy capabilities. Evolution never occurs in a straight line. It takes what's there, it mixes and matches, it improves on it slightly. It builds in redundancy and compartmentalisation as a survival technique. This goes for all living things. And it also develops, again, as I've mentioned, a lot of regulatory networks, the ability to provide feedback on how it's going, how it's doing, so it can improve on that process. Objects like galaxies don't do that. They have other methods of evolving. They go through evolutionary processes, but nowhere near as complex as living things such as brains. And I mean, the structure is incredibly complex. If you take a worm, for example, a simple worm, it has 80 different potassium ion channels, the channels that connect neurons together. Just just one for one type of chemical. It has 1,000 different receptors on its cells. It has 300. It only has a nervous system of 300 neurons, and yet it's got this inbuilt complexity. And last of all, the way information is coded in brains, in living things, is extremely complex. A neuron can, as I said, an individual neuron can accept up to, let's say, 10,000 inputs, and it weights those different inputs, and it integrates them, and it goes through a threshold before it releases its output, before it releases its pulse of voltages. Its pulse connecting it to the next neuron. And these pulses or spikes can vary in their frequency. So there's a spatio-temporal component. They vary in space and time, and they code for different things depending on the frequencies. So this occurs just in one particular neuron, this ability to code information. And the combinations, as I say, are almost infinite. There's certainly more than the number of particles in the universe. So that's why it's important to understand complexity of nervous systems and of brains. Unless we understand eventually, again, those networks, we'll never quite get to the bottom of things like consciousness, for example, things like subjectivity. We can never understand them without understanding the complexity underneath. Now, viewers, moving up the food chain, we also come to complex behaviour, complex outcomes in aggregates of animals. We come to ecologies and ecosystems. We come to shoals of fish. We come to flocks of birds, hives of bees, ant nests. We come to herds of animals. Now, their behaviour is different, again, from the components, just as the behaviour of complex cellular processes are different from the behaviour of individual proteins or individual biochemicals. At this aggregate level, there's a lot of people very interested in behaviour because the ability to manage animals, to be able to manage food chains, retain biodiversity is vital for our survival. Now take the case, for example, of the damselfish. This little fish lives in the Barrier Reef, the Great Barrier Reef. It spawns, and 19 days later the larvae mature, and on the full moon they rise. The question was for the biologists concerned with studying the damselfish was how many of the larvae mature? What are the factors? What are the variables that create the proportion of mature larvae that rise up after 19 days? Now, the biologists concerned tried for a long time to use straight linear equations, linear relationships in their models, and nothing worked because there were hundreds of different variables. And no model devised could actually replicate the behaviour, because the behaviour of the larva, the mature larva, was wildly variable. There were wild gyrations. Sometimes a few would rise, a small proportion, sometimes a vast number, most of them. And suddenly the penny dropped because of work done, again, by other scientists in creating more sophisticated models of complex systems, and even this system is complex. And when the appropriate nonlinear equations were injected into the model, the answer did come out eventually, and the problem was solved. Even though behaviour often appears to be complex, as we've discussed, it's driven often by simple rules. And in this case, in amongst all the dozens and hundreds of variables, there are only three that actually drove the process. And those three were the phase of the moon, the turbulence of the water, and the strength of the wind blowing over the water. And that governed the phase change to maturity of these larvae. and it could be predicted from then on. And that's an example of how very vital data, for example on fish stocks or animal stocks, etc., prey-predator relationships, animal populations in general, which go through massive dynamic phases with, say, the introduction of a feral species into a native population, how that can be better understood and therefore better managed, and therefore that will improve our survival as well as the survival of the species under review. As we progress up the chain of life, we come to the major ecosystems, the ecosystems that control our climate and therefore our direct survival. If these ecosystems change too quickly, it can cause radical climate changes. We can flip from a pleasant climate to an ice age, or from an ice age back to a sauna type runaway effect. So there's a huge number of variables at play here. There's the currents in the ocean, there's the flow of the winds, there's the amount of plant material that absorbs carbon dioxide, there's the amount of volcanic activity that occurs. You can even have feedback loops that stabilise the planet to some extent within limits, such as the Gaia effect, where if the earth and water heats up too much, it changes it kills plankton. As plankton die, they emit dimethyl sulfides, which become aerosols in the atmosphere, forming seeds of clouds. More clouds at higher level reflect sunlight and cause a cooling effect. It cools the oceans, which allows the plankton to survive. And plankton is critical to us because it's at the lowest food chain of life, the lowest level of that food chain, on which we all depend. So that's a negative feedback loop. The Earth tries very hard to stabilise itself, but occasionally you can get these runaway effects, and we may be in the process of one at the moment. As you increase carbon dioxide levels, you increase or reduce the amount of sun radiation that hits the Earth that can escape back. That further heats the oceans, causes greater evaporation, and in the process, again, traps more energy, does more heating, causes more heating, and so you get a runaway effect. Again, that may change because that also affects plant growth. Plants absorb carbon dioxide, act as sinks, and therefore this may reverse the process. You may also get a runaway ice age effect, the albedo process. The more ice that starts to form, the more sunlight is reflected. So the cooler the Earth gets, less evaporation, less carbon dioxide is formed, and ice continues to spread. And the freezing effect may eventually cover the whole Earth. So we have a number of these potential flips, and flips that in fact did occur in the past. We have local effects also where climate can oscillate between two states, as happens with the El Nino and the La Nina effect, caused by the heating or cooling of the water in particular areas of the eastern Pacific Ocean, and flipping the climate in that particular area, affecting winds and rainfall, etc., as we know. This can also occur in Northern Europe, the North Atlantic oscillator. So you can get various oscillation effects which are a common factor in all complex systems until you pass a certain threshold, and then they get completely out of control and go chaotic. But all is not lost, because now again there is an understanding that underneath these complex rules, complex aspects of life, exist simple rules which can be managed. And in fact, scientists are in the process of trying to do just that. An article came out which synopses this new optimism about managing the atmosphere, about managing climate, in the 27th issue of New Scientist, July 2002, Masters of Chaos. The article looks at how climate can be managed for the more extreme events, such as hurricanes or tornadoes. If you can understand the complexities and the feedback loops, it's possible, if a tornado is about to hit a highly populated area, it may be possible to tweak, to change the rules, in other words, to just tweak the energies, change the amount of heat falling in a particular spot so that the wind is slightly changed. So you get that butterfly effect, which can impact even a giant tornado. and turn it slightly away from that populated area. The butterfly effect, of course, is endemic in climate. It says that if you produce very minor variations, changes in temperatures or pressures or circulation in a particular area of the Earth, it can change, can amplify and magnify and change climate quite dramatically elsewhere. So the first attempts at mastering climate are being undertaken right now, and that should have a dramatic effect on our quality of life in the future. Viewers, I think we now have a deeper understanding of the true nature of complexity, because it exists all around us and impacts all our lives. So, viewers, we've moved into a different phase of our understanding of the world, of understanding of reality. It's a much more predictive phase. We're starting to manage and take some form of responsibility for the interactions around us and protect those species that need protecting, and also protect those that don't have a good quality of life. So all in all, understanding complexity is probably the key to our future, and it's certainly the key to our values in the future world. I'm David Tao, and you've been watching again the Theories of Everything program on Channel 31. I'll look forward to seeing you again next week.